In this video, we will discuss about spatial landscape of the immune cells in ovarian cancer patients with and without PPP2R1A genes. So, the reason why they use this different codex system with 26 antibody panels, apart from this bulk RNA-seq, because bulk RNA-seq does not have this special information available to them. This is the plan that they used, that they use the multiplex protein imaging using 26 antibody panel, and this system is known as this codex system. And the cell types are shown here. That is, they observed immune cells and their stromal region, and also they check this immune checkpoint, or I think these are the antibodies that are being used in their studies. If you count them, they might be equal to 26. And thereafter they check the cell phenotype, cell density, and multicellular neighborhood. So first they check the B cells uh, in the overall, in the tumor region and in the stromal region, and they are able to find out that the amount of this B cells is increased in the case where PPP2R1A is mutant for all the conditions, that is in the overall conditions, and also they are increased near the tumor and in the stromal region. And similarly, when they check it for the other immune cells, they found that their amount is also increased. It is in the mutant case, the overall cellular density is increased, and this increase is mainly in the stromal region. The tumor regions, they do not see increase that is statistically significant. And once they identified that in the mutant case there is a increase in the immune cells, either in the tumor region and also in the stromal region, then they did the immunostaining, and they are able to find out that in the case of mutant conditions, they see a TLS formations, that is tertiary lymphocytes kind of structures formations occurs, and this can be seen in this panel D, where the markers, that is CD20 markers, CD21 markers shown a presence of this TLS. And in the literature, it has been already published that this TLS could be a favorable thing for the ICB, that is immune checkpoint blockade therapy. And so from the results, also in line with the previous observations that are being published, and thereafter one more thing that they did is this: they analyze the cells that is in the neighborhood of the tumors, that is around 80 micrometer from the center of the tumors. And when they analyze these cells, they are able to see that the PD1 positive T cells are mainly increased in the case where ICV treatment are been done, both for the mutant and also for the wild type. It can be seen here that before the treatment, the amount of this PD1 plus CD8 plus T cells is low, but after or on treatment of ICV, they see an increase in the PD1 plus T cells, both for the wild mutant and for the wild type. And they check it for the other CD8 plus T cells also, such as CD4 plus, RO plus positive cells, and PD plus, CD8 plus T cells, and CDN. So this panel F is for the one patient and panel G is for the another patient. And the immunostaining images is shown in the H. Here also we can see that the panel F in the panel H is for the mutant conditions where PPP2R1A is mutant, whereas panel I is for the conditions where PPP2R1A is positive. We can see from the visual inspection that PD1 positive cells are low compared to the case where PPP2R1A is present in PPP2R1A is in mutant conditions. The PD1 amount is high in that case. This indicates that the PPP2R1A somehow attracts these immune cells near the tumors, or after this treatment of the ICB, the immune cells get attracted towards the tumors and also in the stromal region. This is what they find out about the spatial landscape of the immune cells in the ovarian cancer with and without PPP2R1A on treatment with ICB, that is immune checkpoint blockade, and how it is different from the case where there is no treatment of immune checkpoint blockade. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.